Okay, cool. Hello everyone. Uh, uh, am I audible in earphone?
Yes, yes.
Okay, cool. Then I'll start. So, hi everyone. I am Gati. I am a BSMS fifth year student in the lab of uh, Dr. Yashra Chauhan. Our lab is called CLUE, Chauhan Lab for Understanding Evolution, and our lab does experimental evolution in microbes, ranging uh, a lot of questions like, for example, evolution of multicellularity, evolvability of phenotypic plasticity, even bacterial ecology, coexistence of species using phenotypic plasticity or multicellularity as a tool. So, my project is about phenotypic plasticity mostly, and we use the macroscopic multicellularity in Staphylococcus aureus, which happens to be plastic, just as a tool to understand the evolvability of plasticity. And this project actually came into being when I was in my fourth year, and Yash was taking a course about evolution, and he was introducing phenotypic plasticity to us and polyphenisms, where he was talking about how there can be different uh, phenotype in different environments, but the genotype does not change. So from that class, we discussed about Waddington selection. I'm just talking about this on the first slide because I have not included Waddington in my presentation today because that would go in, on other direction. But what Waddington did during 1950s or 1960s is uh, he took Drosophila, and there used to be a heat induction. Basically, you give it a heat shock for 40 seconds, and then during its developmental stage, basically, and then it does not produce patterns in its wings. So there are no veins in its wings. So either if you do not induce heat shock, there is veins in its wings. If you induce heat shock, there is no vein. So that used to be called cross veinless. So, what Waddington tried to do is he tried to select under induction both the crossben and crossbenless kind of phenotype, because the phenotype is anyways a proportion of the population. Most part of the population would be crossbenless, but some part of the population would still show those crossbens. So you can still select them and have a frequency dependent selection, as in you increase their frequency over multiple generations. And he tried to see how plasticity can evolve in both in multiple directions. So that was the uh, inspiration for the study. But because there are many people who might not be familiar with what phenotypic plasticity is and what multicellularity, especially in the case of bacteria, is, I'll give a brief introduction. Before the introduction, I would want to just reiterate that this project is not entirely done by me. I have done most parts of it, but it has also been carried out by Robinson. Robinson was a French exchange student from ENS Paris, and the obviously the conceptualization is by my PI and us together, Yashraj. So I'll mention wherever Robinson's work comes up that it has been done by him for transparency. So to start with, what is phenotypic plasticity? Phenotypic plasticity is basically the ability of a single genotype to express different phenotypes in contrasting environments. The genotype does not change, but the phenotype changes. For example, water fleas have a different head morphology with respect to presence or absence of predator. Butterflies have different wing patterns in different seasons, and butterfly pupae can also have different color in response to the background they are in. They can match the background. These are some easy or common examples of phenotypic plasticity that we see around us, and phenotypic plasticity in terms of graph or graphically can be represented by reaction norms. Reaction norms is basically phenotype on the y-axis and environment on the x-axis, and across different, the environment can be discrete, the environment can be continuous, but across different environments you'll have different phenotypes. Now, such a plastic phenotype was discovered by Yash, my PI, where he could observe that both gram negative and gram positive bacterium form multicellular cluster in high salinity. Basically, when you increase the salt content in LB rich media, that is, you add 5% weight by volume of NaCl to LB already, the bacteria does not make the medium turbid, rather makes macroscopic multicellular clusters. How do we know there are bacteria in these clusters? We fish out the cluster, we wash them with fresh media, we vortex them in fresh media and then plate it, and we still get bacteria. So we know that these clusters definitely have bacteria. So what do I exactly mean by multicellularity? So, according to the book Evolution of Multicellularity, the definition of multicellularity is context dependent. If the context is the distinction in life cycle, multicellularity can be aggregative or clonal. If it is in response to environmental signals, it can be obligate or facultative. And if it can be simple or complex in response to the number of cell types and the complexity in size, function, dimensionality, etc. In our case, the multicellularity that we are observing, we assume until now it is simple and undifferentiated multicellularity, although there are experiments going on by PhD students in the lab to look at if to entrench if any differentiation is present at all between the cells present at the top of the cluster and bottom of the cluster and other areas, to see if we can entrench any pre-existing negligible amount of differentiation. But until now, we assume that they're simple and undifferentiated. And conducting plasticity studies is generally challenging, and in especially in such a regime. And to conduct studies which understand evolution of plasticity is even more challenging, because you have to start with this same clone every time. The genotype, you have to make sure that all across all your regimes, all your selection regimes, the ancestor has the same genome every time you start with a selection experiment. And plasticity can evolve in multiple ways, and one such way plasticity can evolve is canalization. What is canalization? So, canalization is the ability of an organism to express the same phenotype regardless of the variation in environment or genetic background. So, basically, it is also called as developmental robustness. So, I have a question for people in the audience today. Do you think canalization is plasticity, or canalization is the opposite of plasticity? Because plasticity is expressing different phenotype in different environments. Canalization is expressing the same phenotype regardless of the variation in environment. So what do you think? Anyone can answer if canalization is plasticity or opposite of plasticity. I can't see oh, there are charts. Can I see? Okay, so canalization is opposite. Actually, uh, canalization, obviously, in terms of reaction norm, it looks like this, that the phenotype is constant across environments. But to be precise, canalization is plasticity, but at a different level. So at the level of structural genes or the trait that you're looking at, the phenotype is constant across environment. But at the level of regulatory genes, there is still plasticity. So regulatory genes would still adjust according to the environment to bring in this constancy at the level at. So you are looking at a global level where you see a constant phenotype across environment, but in terms of the gene regulatory networks that are involved in this pathway, there will still be some plasticity in response to which environment they are in. So when you achieve complete canalization, it leads to genetic assimilation. What is genetic assimilation? Basically, your ancestral population, when you start an experimental evolution, would show some change in phenotype across environments. That means, in terms of this reaction norm graph, it would have some positive or negative slope. But after your experimental evolution, entire selection experiment is over, your evolved organism or evolved strain will not show any variation in the phenotype. So basically, it will have a zero slope. So such change in slopes of reaction norms after experimental evolution of plastic phenotypes is called genetic assimilation. Genetic assimilation is basically complete canalization of an existing trait. And in the case of Yash's study, what he did was he kept on selecting these multicellular cluster for 50 days with gradual reduction in salinity. So every 10 days, he would reduce 1% salinity. So by the end of 50 days, uh, they could see that multicellular clusters also evolved even in uninduced condition, basically this environment, even in uninduced condition via this experimental evolution or this selection experiment. So basically, they saw that such a plastic cell clustering is evolvable. So it might be a little bit much, so I will simplify it. They saw that bacteria can become macroscopically multicellular in high salinity. They tried to select for such a phenotype in over 50 days with gradual reduction in salinity to see if they can fix this phenotype irrespective of the environment. So it does not matter if you are in high salinity or low salinity, you still will be able to produce these multicellular clusters. That is basically the canalization of this trait or genetic assimilation. And they saw that, yeah, that is happening. That means this phenotype can evolve in multiple directions or at least one direction. So, taking this as an inspiration, we asked a question that what if we select for different things? As in, not just multicellularity, can we select for unicellularity while forcing them to become multicellular, or while inducing high salinity, can we select in the opposite directions? In what all directions can we select? Basically, if we are to design 50-day experiments using such a setup, we can select for the multicellular cluster in high salinity. We can select for the unicellular cells which might be present in the medium, which are present in the medium actually. Not all the population, not the entire population is contributing in making this multicellular cluster. Some are still present in the liquid medium. And in the normal habitual salinity, we can also select for unicellular cells because we can fluctuate environments on every other day, which we'll come to later. And instead of selecting for either multicells or unicells, we can also have pseudo-natural selection. Basically, we will vortex the entire tube and we'll do blind selection. So it is like a natural selection itself, but it is happening in lab environment. So it is called pseudo-natural selection. So we designed six regimes in such a setup. First regime is we would do pseudo-natural selection in fluctuating salinity. Basically, we will fluctuate salinity every other day between high and habitual. That is 5% and 0% salinity, and we will select blindly. Then pseudo-natural selection in high salinity, where the salinity will be maintained constant at 5% for all 50 days, and we will select blindly. Then a regime where one day we would select for multicellularity in high salinity and next day we would select for unicellularity in low salinity. This is very complex. Don't focus on it now. I'll come to it later. And the regime which I personally wanted to do like very enthusiastically is selecting for unicellularity while inducing high saline environment. Like the bacteria will get a signal to make the multicellular clusters because it is in high salinity, but I will select in the opposite direction. Then I will do the same thing in another regime, but I'll fluctuate the salinity every other day. And as a global control to all the five other regimes, we will do a blind selection just in habitual salinity. We will not change the salinity level to high salinity at all. And in low salinity, we'll keep on selecting for basically do a blind selection, not for any particular phenotype. So these regimes are named differently and might be confusing. You can switch on your mic and ask whenever there is a confusion. So for all, and I will go through all the regimes one by one. For all such experiments, we had two kinds of media, LB media and high salinity media, which is basically 5% weight by volume NaCl added. And we start our all our selection regimes or all our experimental evolution experiments with a single ancestor. That is, we take a plate of ancestor, we take a single colony and we grow it for 24 hours and then we freeze multiple tubes of it. So I have like 10, 12 tubes to start with. And all our selection regimes can be started from this cryostocks. Basically, you flash freeze them in liquid nitrogen. For those who don't know what cryostocking is, you can store bacteria at minus 80 degrees for as long in experimental evolution, as long as you want, by adding some glycerol, which is a cryoprotectant, and storing, flash freezing them in liquid nitrogen and storing them in minus 80 degrees. So I will go one regime by one. Please stop after every regime if you have any question, or in between any time, because this can be thoda sa too much information at some point of time. So this is the first regime, which is called HI, that is constant high salinity, but we select blindly. We select pseudo naturally. How does it start? Uh, Let me annotate. It starts with just 15 ml high salinity media, where we inoculate 10 microliters of ancestor, and then we grow them for 24 hours at 120 rpm and 37 degrees centigrade. And then they show such a phenotype where there are unicellular cells in the liquid medium, and there is also multicellular cluster here. We take such a tube, we vortex it so that this cluster breaks as much as it can, and there's a homogeneous mixture of unicellular cells and multi cells which are contained in the multicellular cluster. And we transfer 20 microliters blindly into fresh media, allow them to grow again, and repeat this step for 50 long days. And every 5 days, we would store whatever time point, like whatever point in those 50 days would be there, like the fifth day and the 10th day, some representative bacteria from the culture in a tube, and cryostock it in an Eppendorf and cryostock it. The selection was done by Robinson, and I will show you the results in a bit. So the ancestor in high salinity looks like this, but after 50 days of evolution, the evolved bacteria in high salinity, the multicellular cluster formation was even more like it became adaptive. The cluster formation became adaptive in high salinity. So some more pictures across multiple replicates. So this is how the ancestor looks like in low salinity and in high salinity, and this is how different clones and different replicates look like in high salinity and low salinity. And as you can see, the bacteria has gotten the ability to make more intricate network-like looking structures, more intricate threads. Sorry, there's some issue in how I speak. So we use this assay called multicellularity index in terms of optical density, where we would measure the OD of just the unicellular medium after growth in 20 of 24 after growth of 24 hours, and then vortex, and then measure the OD again. So multicellularity index is given by OD after vortexing minus before vortexing by OD after vortex, normalizing it by the OD after vortex. And Robinson found out that in 25 and 50 days of selection, we could see a significant increase in the multicellularity index. That means in high saline environment, that means the bacteria has adapted in high salinity environment, and forming the multicellular clusters is helping it grow better. So when there is a pseudo-natural selection in high salinity, multicellularity wins. That is the first result that we got. The second regime is called fluctuating indifferent selection, which was also done by Robinson, where we would start with high saline media, inoculate some bacterium, make them grow for 24 hours, then vortex it after 24 hours and transfer 20 microliters blindly, but not into a high saline media, but to a low saline media. So we'll fluctuate the environment every 24 hours. That was the goal. To see how the bacteria would be able to evolve in such a regime. So every odd day it will be low salinity, and every even day it will be high salinity. So we'll transfer it to a low saline media. We'll allow them to grow because in low saline media they grow as unicellular bacteria only. And after 24 hours, we again transfer it to high salinity media. And we do this for 50 long days, and we cryostock again every 5 days, and we do contamination checking also every 5 days. So in such a case, what are the results that we see? The ancestor in high salinity looks like this, and the evolved strain in high salinity is forming even more intricate networks of threads of structures, and across replicates. I am yet to take multiple replicates, pictures of multiple replicates, but this is how it looks like. You cannot see the intricate network structure here because the entire network has sank down because of its weight. But when you like try to swallow the tube and then click the picture, this is how it looks like. So this is basically how we do macroscopy. So in this regime, we also saw that in fluctuating indifferent regime, we also saw that uh, the multicellularity index according to at least OD increased. So in both high salinity indifferent and fluctuating salinity indifferent selection, the multicellularity index increases every time you select blindly. So you're not selecting for multicellularity, you're not selecting for unicellularity, but even when you fluctuate the environment, or even if you keep the environment constant, that is high saline environment through 50 days, in both the cases, you see that multicellularity wins, or multicellularity is more adaptive, right? So in that scenario, I had a curious question. Why can't we try selecting for unicellularity rather multicellularity in high salinity and see if unicellularity wins even when we have a strict artificial selection in its direction? So that this regime is called HU, where we inoculate the bacteria and allow it to grow for 24 hours at 120 rpm. Then we select 20 microliters of the unicellular bacteria only from the liquid medium, actively avoiding the multicellular cluster, into fresh media, fresh high salinity media, and allow them again to grow for 24 hours, and repeat this process for 50 days. And obviously, all the selection regimes have five independent lines, which are represented by five different glass tubes. And we cryostock every 5 days, similar to other regimes. So, using such a macroscopy setup, we could see that the ancestor in high salinity, which was almost transparent in its liquid medium, and would the number of cells in the liquid medium was also very low, looked something like this after 50 days of evolution. A very stark difference between the ancestor and the evolved was observed in this regime. And this was also constant across different replicates. Like there are five replicates, right? But we saw that most of the replicates could not lose this potential to form the multicellular clusters, maybe because that is very, very adaptive, as we saw from other regimes, that losing it would be difficult. But they definitely reduce the number of cells which are found in this cluster, because look at the optical difference in terms of macroscopy itself. Like the ancestor is so transparent in its liquid medium, but the evolved strain has almost completely utilized the niche of the liquid medium rather than just forming a macroscopic multicellular cluster. And this is not just in terms of clusters, not just in terms of macroscopy. We also did colony counting, CFUs. And okay, let me just show. So each replicate also has five clones, which we have isolated from the 50th day tube of each replicate. And for replicate one, which was most successful right here, we have five clones, and we almost see that all the five clones of replicate one are very, very successful in becoming unicellular. And in terms of colony counts, also we see the same, where the total cell number has increased in high saline environment as compared to the ancestor across all the replicates. That means they are now more they can now grow better in high saline environment, which is very, very intuitive because they were selected in high saline environment for 50 days. And but to our surprise, that they could also adapt to habitual salinity environment, an environment they were never exposed to in the 50 days of selection. So across 5 replicates, you can see that there's a significant difference between the total cell counts in at least 4 out of the 5 replicates as compared to the ancestor in terms of total cell count. So that means in both habitual salinity and high salinity environment, they could grow better, right? Now we discussed about the reaction norms and their slopes. So our goal was to use these colony counts to plot those reaction norms. So basically, if you have forgotten, let me refresh your memory. Reaction norms were something like this, right? So our goal would be to look at like the ancestor would have a positive slope if this is multicellularity index. The ancestor would have a positive slope where this is low saline environment and this is high saline environment, and the ancestor would have almost zero multicellularity and the in low saline environment, and in high saline environment it would have some positive multicellularity. But after 50 days of selection in this regime, the slope should look something like this, right? I'm dotting it. I don't know how. The slope should look something like this. So the slope should have a significant difference in the reaction norm of the ancestor and the evolved should have a significant difference in their slope. Right, that was the goal. So we tried to plot these reaction norms using this assay called multicellularity index assay using colony counts. So what we would do is we would allow them to grow for 24 hours, then only salvage the unicellular liquid medium and plate them, which would give us the count of unicellular cells, then vortex the tube and then plate again, which would give us the count of total number of cells. And the multicellularity index would be the total cells minus unicells by total cells. So we normalize it by the total cells as well. So using such an assay, we tried to plot reaction norms where across, so these are the clones of all the replicates. We could see that in replicate 1 and replicate 4, there was a significant difference in slope in the evolved strain as compared to the ancestor. That means, except for replicate 2, 3, and 5. But in terms of macroscopy, you can see that there's a significant difference in terms of how they are growing in the high saline environment, but in ancestor versus the evolved. But we could not see that stark difference in terms of multicellularity index reaction norms. That means that it is not very easy for all these bacteria to lose out on multicellularity because it is also very adaptive. So that means it is very, very difficult for them to become completely unicellular. So they do not completely give up their ability to form multicellular clusters. And only replicate 1 and replicate 4 showed some significance. So we were not happy with this kind of an assay, and we tried to change the assays, which I will come to in a bit. We from CFU assays we went to microscopy based assays, which I'll come to in a bit. But we also did the whole genome sequencing of the clones, evolved clones in this regime, and we found that we saw clean mutations with more than 80% confidence or 90% confidence in several clones in proteins like FNBA, which is a fibronectin binding protein, penicillin binding protein, which is an uncharacterized protein in Staph aureus, lysae protein, and some translation initiation factor 2. Instead of translation initiation factor, all the other three proteins, and there are few more also, which I am yet to do the analysis and find the annotations. But all of these three proteins are outer membrane or surface proteins, and they have been reported in several literature that they help in cell to cell sticking. So if there is a mutation in proteins which help in adherence or cohesion between cells or adherence to glass tube or a different kind of cell, that means they might have led to less clustering or the mutation could have also led to better clustering. But because this is a gram-positive bacteria, we do not have any uh, previous data on this, like our lab does not have. So we have to look at it, like we have to do a lot more analysis using this genome sequencing. Like we can use AlphaFold to look at the folded structures and everything, how it changes between the original one and the mutated one, and find out at exact what position of the protein there is a mutation, and how it can lead to better clustering or lesser clustering. That is still yet to be done. So this was one regime which I undertook. The next regime also, which is very funnily called FU, was undertaken by me in this experiment, in this project, where basically we select for unicellularity, but we fluctuate the environment every day. So we start with high salinity. We allow them to form these clusters and become unicellular. And after 24 hours, we do not select the multicellular cluster. We only select the unicellular bacteria into 15 ml fresh media, but not high salinity, this time low salinity media. And in low salinity, they'll grow unicellularly. And after 24 hours, we take unicellular bacteria from low salinity again into high salinity media. So every day we'll fluctuate the environment, but we'll select only for unicellularity. And we did it for 50 days again, and we tried to cryostock every 5 days and check for contamination. And such a regime, we saw that the ancestor in LB looked completely turbid, but very counterintuitively, when we selected for unicellularity in fluctuating environments, the evolved bacteria could also become multicellular in LB. Very, very counterintuitively. So you would never expect the evolved bacteria to become constitutively multicellular, but such is the curious case of adaptive plasticity, that if at all a plastic phenotype is very adaptive, it would get constitutively expressed irrespective the direction of selection. So even though we selected for unicellularity, somehow it could also become multicellular even without induction. That was very surprising. And in high salinity, the ancestor looked like this, and in high salinity, the evolved look like this. So this is also very, very intuitive, because we were selecting for unicellularity. So we would expect that the bacteria would explore the liquid medium or the unicellular niche more. But according to the HU regime as well, they have not completely gotten rid of the potential to form the multicellular cluster, but they can explore or acquire this niche of the liquid medium as well. They can grow unicellularly as well, better than the ancestor. But they could also become multicellular in LB. Very, very surprising. And this is across replicates. I'm yet to take pictures of multiple replicates, and the it is very symmetric across replicates, unlike many other regimes. Uh, and this is the FU regime. And finally, in the most exciting regime, according to me, is LI, where which was supposed to be a control when we started out with. It was supposed to be a control, right? So in LI, it is very simple. We take 15 ml low salinity media. We grow bacteria in it for 24 hours. They grow unicellularly. We vortex vigorously so that even if there is some multicellular cluster, we do not select it. And then we select 20 microliters into again low salinity media, because we have to maintain the low habitual salinity. We allow them to grow for 24 hours again and repeat this process for 50 days in this experimental evolution. So what do you think should happen at the end of 50 days in such a regime? When you keep on selecting for unicellular bacteria in a medium where there is no high salinity induction, so multicellular structures cannot be formed. So what is your guess that what will happen at the end of 50 days? You can type also. Yeah, should remain unicellular. That's just Anand, because it's Anand, because Anand knows it. Anand is my friend. Anyone else would like to take a guess? Uh, People from Fisher's Wishes or juniors who have joined. At the end of 50 days, what should it? Okay, let's assume that you're also thinking it should remain unicellular, but to our surprise, the ancestor, after pseudo-natural selection and habitual salinity for 50 days, could form mats. So mats, liquid air interface mats in Staphylococcus aureus has almost never been reported, especially when they're grown in a shaken environment at 120 rpm. But the selection in habitual salinity for 50 days could lead to the evolution of a liquid air interface biofilm in Staphylococcus aureus, which is an immotile bacteria under shaken condition, which was the most, according to me at least, the most interesting result in the entire project, because they were unable to form any macroscopic structures at the start in the ancestor, but after 50 days of evolution, they could form liquid air interface biofilms. Now, why that is important? Because biofilms are generally formed at the liquid air interface because oxygen is high there. And because it was shaken, most of the bacteria were allowed to be mostly when something is shaken, a tube is shaken, there is a liquid column, liquid air column formed. Most of the bacteria would try to be homogeneously distributed in the liquid itself. But still they could grow at the liquid air interface and form a mat in low salinity. And across low salinity and high salinity, you can see the evolved ones have their ancestral plasticity, that these thread-like multicellular structures, they are still being able to form because they were never selected against, or they were never exposed to any selection. These thread-like structures, they are still being formed like ancestor. It's very similar, but in low salinity, they could form this liquid air interface biofilms. That was the most exciting result from this entire project. And for the final regime, which was
Hey,
completely conceptual.
hello, is
Yes.
it audible? Yeah, I had a very quick question. Did you see the appearance of this particular mat-like structure in high salinity evolved?
No,
No,
we
okay,
didn't see such intricate mats and such thick mats as you can see here forming in any other regime.
I see.
Mhm.
Interesting. Thank you, Gati. Thank you. Yeah.
So for the final regime, which was completely done by Robinson and also kind of conceptualized by him, which was to select in direction of plasticity. That means on day one, we start with high saline media. We allow them to grow for 24 hours. And we take fish out the a part of the multicellular structure in fresh media, vortex it, and select 20 microliters of those cells, but into low saline media. So we fluctuate the environment, but select for multicellular cluster on day zero or day two, the even day basically. And on odd day, we allow it to grow in low saline media and just select whatever is there, as in the unicellular bacteria. So we select for multicellularity in high salinity and select for unicellularity in low salinity. Now, remember the reaction norm initially. How was it? The ancestor was like this. It was unicellular in low salinity, that is less multicellular, and multicellular in high salinity. Let's say this LS and this is HS, right? So we are selecting for more multicellularity in high salinity and more unicellularity. That is almost like placing a block here. It should remain here and this line should go up. So this should become something like this. The slope should increase basically. So we select for more unicellularity in low salinity and for more multicellularity in high salinity. That is in the direction of plasticity to make it more plastic. Uh, Again, I would ask the question, what would you expect in the end of 50 days would happen? Anyone. So we are selecting for something like this. They would, I would say, your guess would be that they would become more multicellular in high salinity and they would still remain unicellular in low salinity, right? Because the slope should increase. But also, again, to our surprise, they could. become multicellular even in low salinity. And in terms of all the graphs that we have plotted in the OD graphs and CFU graphs, we can see that something like this is happening, where we are trying to increase the slope of the reaction norm, but instead it is trying to maintain the slope and rather becoming multicellular in low salinity as well. So it is kind of having a reaction norm slope inertia, which is what we have termed this reaction norm slope inertia. Currently, we are trying to do microscopy assays. So there was a small problem. I couldn't, just one second. I couldn't, I took some microscopy pictures, but I couldn't show they were in a different format and couldn't be uploaded to Excel. So let's say a microscopy image looks like this. I hope you are able to see my screen. So this is the microscopy after 24 hours of growth. So what we do is we try to count how many clusters are there in each how many cells are there in each cluster, basically. So we'll count using ImageJ how many cells are there in each cluster, and then we'll try to form a image like this, where the cluster is numbered and the number of cells in the cluster are also present. And from such a setup, we will try to plot a histogram, and we'll take multiple images across multiple we'll count multiple clusters across multiple images. So what we'll do is we'll try to form a histogram which looks like this. We'll try to form a histogram of the how many clusters have 1 to 2 cells or 3 to 4 cells or 5 to 6 cells and 7 to 8 cells. We'll try to form a histogram like this, so that what we can do is we can finally have a reaction norm which looks like this across habitual salinity and high salinity. So this is a data that I have already obtained for the ancestor, where the cluster size is generally the median number of clusters across from 300 clusters at least across, I don't know, 300 clusters were sampled from thousands of clusters. Most of the clusters have 1 to 2 cells in habitual salinity, but most of the clusters have 3 to 4 cells in high salinity, but the spread is also different. So the reaction norm looks like this for the ancestor, and we are yet in process to obtain we are doing the microscopy, but to obtain these reaction norms for the evolved strains as well, right? So by doing this, we can have a better idea of how the evolution has happened rather than using CFU counts. So by today, we have completed all the regimes that we started conceptualizing our experiment or project with, all the 6 different regimes. And we can see in most of the regimes, multicellularity is somehow being constitutively, because it is adaptive, it is somehow becoming constitutive across environments, but we are yet to find the basis of it, or at least plot reaction norms to understand the evolvability aspect. But we can show that at least there is bidirectional evolvability between this regime and all the other regimes. So basically, if you want to have an emerging picture from this entire project, we can mathematically try to look at how reaction norms change over time, how their slopes change over time when there are different kinds of selections. So this is a very fundamental kind of project to understand the evolvability of plasticity using a plastic phenotype. So this is not more related to multicellularity, rather more related to plasticity. We just use multicellularity as a tool. So that is the emerging picture. And with that, I would like to end the presentation and thank my PI, my lab members, Robinson, my co-worker in the project, who is now in Pasteur Institute, Anurag, Hadi, Pratyay, Vignesh, Kalyani, Shiva, who is now in Barcelona, Dia, who is now in Sweden. And with that, I'll end and I'll take questions if you have.